So first of all, I want to thank for invitation to give this talk on this nice workshop. And so let's start. So I want to talk about cellular image data. And when I thought what I want to present here, I was a little confused because in reality, mostly we are facing opposite problem, like say inverse problem. We don't have the data we want to visualize. We have visualized data somehow, and then we want to understand what is behind. So this is like inverse problem. And the generally inverse problem in many cases in the engineering and in science always more complicated than direct problem. So for example, if you know what is radio, you can draw how will look like radio inside. But if you see radio inside, is it so easy to draw the principal schema, how it's connected, what is amplifier, what is here, etc.? It's not so simple. So the same, let's say, present here in my talk these things. So what is our main goal? So our main goal, extract functional information from the data which originally are descriptive, which are imaging, and then present it back to the people to inspire idea and to understand what is going on here. So it means we take something which in the imaging, in the microscopy, originally visual, so it is visual. Then we have to convert it in some way and then present in some other way which will be suggestive and allow to understand something which we don't see originally. So to present these topics, I will go to image analysis. I will tell a little about visualization of quantitative data. And then I will touch at the end multidimensional data presentation. So during a long time, cellular visualization and microscopy was, in a sense, descriptive. And up to now, the descriptive part is very essential. In many papers, you find out the images which are like this, where people just with small arrows show, look, this on this here. Or even you go to this super resolution microscopy, mostly this is just represent. Okay, you see this is very blurred. But if you go to super resolution, to palm, then you see it with very much structures. So this is just improving or presenting data, these data which are visualized at the beginning. And this is generally, let's say, little old style, classical style, but still quite working style in the microscopy. There is another way when you go, which is where this descriptive, this presentative way, illustrative way does not work. This is when you go to some phenotype or some data which is not like black and white. On previous, I just showed, look, this green dot on this red line, this endosome on this part of cytoskeleton. You see it. On another example, which is normally controlled, you don't see it. Okay, this is black and white, very simple. But could be situation much more complicated. This is little more, and this is little less. And if you start to compare by eye, this is very essential when you look at it together. If you just see it one by one, you can't say, is it brighter or not brighter? Our eye is very adaptive. You see the same color, and you see it second time, and you don't see the same color. It's dependent on what you look before. So we are very adaptive. In this case, we have to go to something more reliable and more quantitative in sense. Another problem when we have to go to more quantitative way, this is high-throughput microscopy, which is now becomes a really big deal. So when you have something like this automatic microscope, it's easy to generate millions of images on the one experiment. So what you will do with this million of images? Even with 10,000. You can't even look through them. Okay, you can maybe spend half a year to looking through them, but on the next day, you will don't remember what you looked at the day before. So it's absolutely unfeasible. So you have to somehow quantify it and extract information. Then it's dependent on which scale of magnification you are working. You are going to different algorithms and different approaches to extract information and different kind of information you have to extract. There is no general approach, let's say, like here. For example, in this example, what you put on left, you see like dots, it's individual cells. So in this case, you see, okay, we put, the authors put the virus in the cells, these two different kinds of virus, And they see, okay, here virus is mostly, this is fluorescent virus, in the middle of colony. But opposite this virus mostly infect edge of colony. So this is very different presentation than if you go here, you see much bigger cell, and you see how cells are organized and how look like boundary between cells. So for example, algorithm which we'll use to present and to recognize this nuclei will not work here on this image. Then if you go inside cell, for example here, then this individual dots is organelles, in this case endosomes, and they are much smaller than nuclei. And again, the algorithm which we can use here on the first case, I don't see my laser myself, you can't use here because in this particular image you will be limited by diffraction, and then your image will be much more pixelated, so the previous algorithm will not work. So what's the general algorithms which people use for the data analysis? So this is example of image, which multicolor image, which done on the automatic microscope. You see the nuclei, blue. You see two different markers, green. This is marker for the endocytic, for the small GTPase RAB5, which is fused with fluorescent protein GFP, and which is marker for the early endosomes, and red is cargo. In this particular case, this is EGF. So this is cargo molecule. So if you want to look on the nuclei are relatively big, what you can use here? Okay, there is very standard approach. You can take the threshold, cut off all intensity below some threshold, then take the things which above threshold and find out boundary. And this will be very straightforward approach. This will be nuclei. Okay, so how to do it? Very simple. You take the histogram of intensities. You see very clear peak. You see this is logarithmic scale, majority on this clear peak. This is pixels which belong to the background. Okay, you decide, okay, this minimum is correspond to transition point. I will put my threshold somewhere here. So how it look like? So if I change the threshold, you clearly see that I go through this peak, and then I start to eat the nuclei. Okay, so I return back, and I stay here. Okay, this is good if I do it manually, but I can't do it manually for the 10,000 images. But the answer to this question is quite old. So in 75, it was offered the first way how to do it by maximization the ratio of variance within the classes to the total variance. And this is on the work of Otso, let's say 30 years old work, how to do it. So what is used now is different modification of the Otso approach. They just change the function to maximize or minimize to divide the intensity on the classes. So you divide intensity on the classes and say, okay, this is background, this is foreground, find out boundary, apply maybe some morphological transformation to find out the topologically connected areas, and this is your data. Maybe later apply additional filter to remove something or find something which is more specific or not fit to the idea what could be nuclei. Okay, so if we return back to our original image and look on another part of it, so I little change brightness, so you start to see background, and you see, but it's not so simple. Background is dotted too, so this is dotted and this is dotted. It becomes more complicated. So now I zoom in, and then I want to find out what is here on the green channel, on my EGF, on the endosomes. Okay, so this is just, I isolate the green channel. I take histogram, and now I see quite long tail. Okay, I still believe maybe somewhere here, this is my threshold, which I will offer the standard way. If I go here, no. At this point, I saw mostly the wall cell. And at the very end, I still see all this part like wall blob. It's not separated on single endosomes. And if I return here, I have background in the wall cell. Because quite big background staining, I can't isolate endosome in this way. So what I can do in this case? Again, there is very standard approaches which call it adaptive thresholding. In this case, I will do the same approach, but different, but individually for every small piece of image. I divide image on the small pieces, and for every piece, I will do the same. Then I find out individual threshold for every piece and do it this way. And if you look on the majority of paper which published now, they use every variation of this approach. So in this case, you can take the image, but, problem is that endosome, what we look for, is very small. So this is pixel size, and pixel is less than pixel is about 100 nanometers. It's quite small, it's below diffraction limit. But how many pixels belong to the endosome? Where we put the border? Here, here, here. So then depending on how we choose the border, then we have very different answer. And all our results will be very discrete and pixelated in the level of the quantity we want to extract, size of endosome, amount of fluorescent carga, etc., etc. So how to handle with this? So there is another approach. When we use, we start to use it from 2003, I think, and published 2005. When we start to feed the image by set of base function. So we use the base function, which in reality head-like function, and we describe the image like sum of individual functions which simulate image. In this case, we are not limited by number of pixels because our function defined in the continuous space. In this case, we describe in continuous space our image. Okay, then if we do this, and this is example how it's look like. This is original image. The gray scale, you see intensity of original image. And red, you see the result of fitting. It's a little shifted on Z because I don't add background here, which is another parameter of fit. So this is how procedure look like. It finds one by one image, and this is part of visualization, which help person to look that system works properly, and start to remove endosome one by one from original image. So at the beginning, you saw quite clear difference between the cell and environment around. But after the whole endosome and the whole signal will be modeled and removed from the original image, it's hard to say where was originally cell. So at the beginning, you saw cell very easy, but at the end, you don't see it, and everything becomes like homogeneous noise. This is visualization in sense, and this is control at the moment when you adjust algorithm to see that you really find all the object in the image. So what you have at the end. So on the left, you see original image. On the right, you see the image which I generate, like putting intensity and visualize intensity of the my function, what I found. So this is original image. And what is good here, first of all, what I found here is sub-pixelar resolution. So I don't limit it by pixels. Second, it is high dynamic range. So the dimmest object here, which found on this area, is 2 orders dimmer than this one, and all of them are captured. I'm quite sensitive to the relatively high background from the fluorescent protein, which is not bound to the endosome. So everything works more or less well. So in 2009 was offered another approach from the Zurich University that which use the active contour on sub-pixelar resolution when you put artificial force to keep it. You know the idea of active contour, so maybe I will not go to the detail, or I have to. So this is a question which I asked to auditorium. So auditorium don't answer, it means they need some explanation. So idea of active contour is very simple. You introduce artificial contour, which has forces inside. You model it like physical entity. So it has forces inside. One force try to shrink or expand it. Another force resist to bending. So you have some rigidity of this contour. And external force produced from the original image. So generally, this approach is used on the image which is not pixelated, when you don't feel pixels yet. In this case, you easily can calculate like external forces, intensity, or gradient on original image. And then you allow to this contour to evolve until it stays at some point when all forces will be balanced. So in the minimum of energy. So the forces which are from the image are fully balanced by forces of the contour. So this is called active contour or snake. In this particular case, approach was applied to the subpixelar things by interpolation of images in between pixels to generate the force from the image in between pixels. And they show in reality that they can find out the endosomes. They work with the same RAP5 endosomes with much higher accuracy with subpixelar resolution. So at the moment, this is only two approaches which allow to quantify in a good way in the high dynamic range images like endosomes, which are sub-resolutional. OK, we quantify it, and then now I switch to the next topic, and we extract information, what we have to do with it. OK, I take integral intensity, which is sense proportional number of fluorescent protein on the endosome. And I want to see the distribution. I just make the histogram, and I put in every bin how many endosomes I see on this histogram. And this is generally what I see. You see, the whole tail in reality is here, but this is only few endosomes with this intensity. So I don't see it at all. And I have everything condensed on the left side. OK, maybe it's better to draw the x-axis in logarithmic scale. Yeah, it's a little better, but not much. Again, everything here you have huge bins, huge, they are the same, but in logarithmic scale, they look like huge. This is, again, problem of visualization. And everything condensed here. OK, maybe in this case, better to present this if we bin our histogram in logarithmic scale. What we will have in this case? This is what we will have. So now you see really distribution. You see how this is distributed. You have even idea that there is like two pools. One is here, and another is look like here, with different property, which we can try to analyze. But we immediately face with situations that if you use standard toolbox, they don't work with the histogram, which have not equal bins. They all assume that data histogram represent density. But this is not density, because every bin is different. And this bin is, let's say, 2 order of magnitude smaller than this bin. This is point of visualization and presentation of data. But specifically presenting in this way, you extract more information. Now, let's go again, continue with this presentation. Suppose that I want to present, and this is, again, point of visualization, how connected different property of the endosomes. So we quantify endosomes, we find out size, we find out how many proteins on every individual endosomes, and we want to understand how they are connected. So what is standard way? I do scattering plot. I draw radius of endosome on one axis. I draw the amount of the density of RAP5 on another axis, and I draw scattering plot. If I just draw scattering plot, I see virtually nothing. OK, the next step, I start to color coding the density of the dots. So the red is, you have the majority of density, the green is less, and blue is most sparse things. And again, what you see here, yeah, I will say, hard to conclude, yeah? But if I present this in a little different way, I take every individual bin and average it on the intensity. In this case, I see quite interesting curve. So I see dependencies, that mean value grow along the radius. So with increase of radius, it's grow and then start to fall down, at least this point significantly down. So this is very different. But what does it mean? If on top of this, I will take the movie time lapse, and I will see how the individual endosome progress, and add here some time information, which here I draw by lines to show the direction of time development. On the different experiments, they provide a different slope, and then average the experiment. This is what paper was published in 2005. Then I understand that, yes, in reality, endosomes, the RAP5 endosome progress from the small dim guys to the big and bright guys, and then this is collapse. And this collapse of RAP5 has very big biological meaning. It means progression from early to late endosome. So to the people who are not biologists, I explained that the progression of cargo, or it could be nutrients or signal inside the cell, they come from outside, go in the endosome, the first station, early endosome, where I sorted, and decision is done. Which has to be recycled back, which has to go to degradation. and the cargo which has to go to degradation have to be delivered to late endosome. And late endosomes, they partially degrade and then delivered to the lysosome where fully degrade and reutilized. So this is point that this was shown that early endosome going through the growing cycle of Rab5 and then Rab5 disassemble and going conversion to the late endosome, which was quite big impact and quite big significance finding. Another point, if we go to the same endosome, as I told, endosome are motile, and this is another presentation, this is live cell imaging, you see how they move. So again, we can quantify endosome, we can do the point of tracking and find out the fate of individual endosome, but what we can do with this after that? Okay, another way of presentation, we want to understand, is Rab5 really regulates the protein, which we follow, regulate motility? Could we do this? Yes. We present the intensity versus speed. So on this axis, I draw speed, and this axis, I draw intensity. So if you see on this graph, is it suggest you something? I will say not much. Okay, I understand they have somehow distributed. You have somewhere a peak, okay, but maybe it will be better if I start to rotate it. For example, to see it from different side. Again, yeah, I see this is maybe not very symmetric, but does not tell me much again. But if I do little different presentation and start to, it does not play, sorry. Okay, so, and start to make different cutters of the original image. Then I see the situation is not so simple. If I move along the speed, I have very different distribution of the intensity of endosomes. It's not only going up and down, it qualitatively changes. At the beginning, it has peak on the big side, size of big endosome, and at the end, very high speed, it going to the small endosome. Just looking on this presentation, I have inside that in reality, the Rab5 has negative regulation role on the speed of endosome. So if you go from the slow endosome to fast one, you go from the bright endosome to dim one. So this shows a negative regulation role of Rab5. And this is suggestive from the data, which we have originally. But this, sorry, you can conclude neither from this movie. If you just look on it, this is the original. Okay, it doesn't play. Okay, it doesn't matter. You understand the movie, what you saw. And this you don't see from the movie, what you see here. So this is another thing which you have to take, another data presentation. So now we can extract a lot of data. And this lot of data which we can extract, this I just list multiple data which we can extract in the different screen. And if you put all of them for all images, you go to the huge table like that, when you have a lot of parameters with different conditions. But what you do with this table? So then we go to the idea of the profiles. So for every condition, we start to draw a profile when we say, OK, divide all our parameters on the group. The group depended on the one, for example, cargo, in this particular case, EGF, to another cargo, transferrin, another group of parameters. And for every group, we say, OK, this is change of the number of endosome. This is on uptake of cargo. This is cargo density, et cetera, et cetera. So we have explanation of this. For the one profile, this is very good. But if I put multiple profiles on top of each other, then it becomes not so clear. So for example, here I see that profile very much fit to each other, and this is good. Here I see they are not much fit to each other, but if I remove something which is not fit, then I see, OK, these two of them are really very much resemble to each other. But how to isolate them? One of the ways which is standardly used in the field, this is color mapping. I can't say that I'm fan of this presentation, but at least it's allowed easily to see if two profiles resemble each other, if you have to present multiple profiles. But this is, again, limited to the few in reality. If you go to few hundred, it will be hard to see anything. So then we go to the last point, to the clustering. So the next way how you can combine this, you say, okay, I don't want to see individual profile, I want to cluster them to the group. And this is another presentation of the clustering we saw today in multiple talks, something like this. And then we go to the point that we think about presentation, but not so much how we cluster. But in reality, if you look on this, in the field, at least of screening, in the last publication, you see that in reality, people use very few clustering tools. So hierarchical cluster is winner. The second one will be k-mean or different combination of k-mean, phase c-mean, etc. Up to now, people use manual clustering, so which a lot of work, but in some sense maybe not so bad. And this is only one when we use the density-based clustering. But if we look on this, how much we win if you use different of these clustering approaches. So this is example which we try to understand if we cluster experimental data of genome-wide screen on the endocytosis and try to apply to this different clustering tool. So what I draw here. So on this axis, okay, laser, I don't see, so it's dead. Okay, on the left axis, you see number of clusters. On the right clusters, you see number of clusters, percentage of clusters enriched in the protein-protein interaction. So in reality, if you take the screen, and if your data are meaningful, of course, your cluster have to be enriched in the protein-protein interaction, because at least part, besides its noisy, etc., at least part of them have to be because of protein-protein interaction. So what we see? So we see that in the standard clustering, which is hierarchical clustering, we see very little enrichment in the protein-protein interaction in reality. And if you start to decrease point when you cut cluster, because in the hierarchical clustering you have the full tree, then you have to make the separate cluster somewhere, cut it, and decide, okay, here I cut it, and this is my clusters. If I start to cut it too low, I have a huge amount of cluster. What's the sense of these clusters? If you go to mean shift, the number of, to k-mean, the number of clusters is not grow so much. It's okay, it's linear in this case, but still enrichment is not so big. The density-based clustering behave much better. So this is, again, I want to put these 2 points that visualization and presentation is essential, but essential what it present. So if clustering algorithm is not working well, does it matter how you present it? If it's maybe only misleading because what you present here, mostly noise. So in this case, I almost in time. In this, I want to finish my talk and say the questions which I still see like open. So how to properly present high-throughput phenotypic data that they will be comprehensive. So clustering is OK, but even in clustering or within the cluster, how properly presented, I still not know. The best what I saw, this is color coding, but you see the number of levels of the color, which we can easily recognize by eye, is not so big. And I don't feel it like quantitative. If you start to draw multiple lines on top of each other, and for 100, it will be a mess. Very hard to understand anything if they are not precisely correlated. So how to present it, I don't know. And another problem, if this is very much connected to the previous one, how to understand what they are similar, how to measure similarity. And this is, again, from my point of view, open question. So I want to acknowledge the people who did the experimental data, which was not published yet, and which I refer in this work, people who helped me to develop the different algorithm and presentation and do some experimental work, and the whole Marina Serial Lab, when I'm working so good the last 6 years. Thank you very much.